So we're going to speak about the Functionnectome application. And as Stephanie explained to you, it is a new method that we developed that is putting in place the idea that the world is more than the sum of the parts. So if you think about it, when you do fMRI, because you assume that people will not have exactly their brain regions aligned together, you add a Gaussian smoothing that will kind of like smoothen up the edges and allow you to have a maximum significance for the one area that does not necessarily overlap in Elizabeth and myself, or you and myself. But if we smoothen in the world of like, I see everything blurry, they might overlap. The same way that if I didn't have my contact lens on my glasses, all your faces would be the same. Okay. And we try to change this prior by being, instead of smoothing up the cortex, we thought like, what if we apply the same structural connection priors to everybody and project the signal instead of smoothing it up like randomly around the voxel, just projecting the signal exactly the same way for everybody. And by doing so, minimizing the effect of blurring, but also putting anatomical constraints and testing really whether the co-activation, the co-variation of the bold signal in the brain is better to model the phenomenon, the activation phenomenon in the brain, okay? And that's what the functionnectome is about. So you take the classical fMRI, you use anatomical priors of how the brain is connected together, derived from high resolution diffusion weighted imaging, best spectrography built by our friend Flavio Delacroix. And then you use a weighted average. So you create this probability of how every voxel is connected with every voxels. And you use this information to do a weighted average of the projection of the cortical bold variation onto the white matter. It is just a transformation of your raw time series bold signal, normalized to give a nice space, into a functionnectome. So it's a transformation of the data. And those data that are transformed will not change the rest of the processing of your fMRI. So you place it inside this timeline of processing where you had your raw fMRIs and you acquired with the MRI scan. You did your pre-processing of denoising and normalizing, registering it to the MRI space. And then you apply the functionnectome. And finally, you do the statistical analysis and the statistical math. Okay?
But you don't smooth them.
But you don't smooth.
Right.
You can smooth if you want, but that will be doubling your assumption and your kind of like normalization of the brain. Do twice, I would like cancel out differences. We did test with and without smoothing. It is better statistically not to smooth. It's my recommendation. And as a proof of concept, to give you an idea a little bit of the difference, we applied this to the simple task of finger tapping. And you can see here a beautiful finger of mine that I draw in PowerPoint, pressing a button. If you do classical fMRI, what you do standardly is have those activation in the gray matter because it is very rich in vascularization and astrocyte. That's where most of the oxygen is consumed, and that's where you see your bold signal with the maximum contrast. And you interpret the brain areas that are activated locally. You compare voxel to voxel. If you use a functionnectome, you will have this. You project the functional MRI signal onto the white matter. You run your statistical analysis, not using a mask of the gray matter only, because otherwise you won't see the gray matter. And instead of having activation on the surface of the brain and subcortical areas, you have entire connections that come out. Now, if you think about the motor system and the finger tapping, it is obvious that it is not a single brain area that is responsible for this. You need to gather the implication of the corticospinal tract, corticostriatal connections, as well as cellular contribution to the movement that you're going to do. So it makes sense to look at circuits more than just single island of areas that are activated. All good up to now? You can interrupt me anytime if you have a question.
What kind of prior you give to for your analysis? The one of the patients of an atlas?
No, it's an atlas based on the Human Connectome project. We do have several different priors as using deterministic or probabilistic tractography. I'll show some details after that. We also have new priors that separate commissural projection and association fibers, because as you can see, when you have your final result, sometimes you have a mix of fibers that intermingle. It's hard to interpret, but if you divide the fiber per class, then you have less crossing and you see clearly the system that is involved. And then if you do use the individual track, so remember what I said at the beginning, every brain is different. There is one way to tackle it is you wear blurry glasses. That's the smoothing effect. Another way to tackle it is assuming that everybody is connected the same. So you project the same way on every network. But if you use individual tractography, then you embrace this entire individual variability, you decrease your statistical significance, so you eventually do single cases analysis of your fMRI. That's what you would do.
Yeah.
And in that case, yeah, you probably have better results. But if you do group studies, then you will lose your effect just because people are too different. Any other questions? So you have no?
Yeah,
Yeah.
I don't feel it's like if you are doing a ROI, you are putting arrows at the cortical level in order to see what is the structural connectivity under this arrow.
This is at the voxel level. There is no arrows. But every single voxel is projected onto the white matter at the raw ball signal, and then you assess statistically the tracks that are significantly involved. So it's not like using a region of interest of the activation or big regions to do the projection.
It's at the voxel level.
Yeah, and that's why doing it at the individual level, like recreating the priors is very long, because you calculate in hundreds of subjects the probability of every voxel in the brain to be connected to each other to create those priors. It is long, but it's feasible.
It's perform univariate analysis.
But you can do any kind of statistical analysis you want. You can do univariate or multivariate as you wish. But the assumption of the association or emergence is coming from the transformation of the data, not the statistical model. So we still do voxel-wise analysis, looking at voxel independently, because we already created this association projecting onto the white matter. Cool. Yes.
To be clear, so we are analyzing some sort of movement or something that the patient did during the fMRI.
That's LC control. It's just doing like finger tapping.
Okay, so the protocol
versus no finger tapping.
Okay.
So when you finger tap, you use more oxygen in these areas. And if you project the variation of oxygen onto the white matter, the weighted average of the projection show you this involvement of connections. Okay. Because the projection, both sides of the projection of these tracks show increase and decrease of oxygen consumption when you do the finger tapping versus when you're not. Oh, cool.
And principally you can perform it on a single subject.
Yeah.
So the group size doesn't really or is it measures statistically, but
Yeah.
you can do it in a single subject.
Everything you can do with the fMRI, you can do it with this because it's just adding like one step in the analysis.
Did you try it with memory, or did anybody?
Yes,
try it with
we,
memory,
I
or
think I have it, but that's a memory from the Human Connectome project. It's not like great memory paradigm.
okay.
Yes.
So in Foulon also do a group level, registry for the group level, or is it only subjects?
You can do anything you can do with fMRI, single, group, different people.
But fMRI should only be time series, so not processed, obviously.
The input should be time series, not processed. Yeah, yeah, yeah. Because otherwise, if you already do the statistical analysis, you did it in a topological way, and then you're just looking at what connect areas that are activated, and rather than testing statistically whether you have a convergence or variation.
Yeah, our work of creating, for example, connectivity matrices. But in this case, you also integrate that with diffusion. Because I'm not
Yeah, so like the connectivity matrices with fMRI will show you areas that are synchronized in the brain. Here we're interested into the circuit in the white matter. Why are we interested in the circuit in the white matter? Because the assumption is the circuit in the white matter is what is defining your function. That's one. And two, by having those maps, now we can directly link together patients and lesion to activation in healthy control. Because we have a comprehensive vision of the circuitry that is not only the cortex, but also the white matter. And of course, based on those maps, if you cut this, you won't be able to move. Based on this map, if you have a lesion in the white matter, it doesn't matter. Yeah, wow, tons of questions. That's good.
Sorry.
No, it's good.
Just we input the time series fMRI data,
Yes.
but what is the output maps we get? It's time series.
It's exactly the same format. It's time series. It's projected time series onto the white matter.
Ah, okay.
Yeah.
So you project like the voxel data onto the prior.
You just have to press run. It will play for you another nifty. Exactly like the one that you put inside, but transform. There is no change in the format, the dimension, anything. It's like, it's the same thing. So what you put in, you get out, and then you can put it in your routine. Sweet. Yeah.
Thanks, Michel. I think Eduardo is doing MEG.
Yes.
For this. So can you basically do any time series?
You can do, so here we use it as an example for fMRI because this is something that we wanted to tackle. But we currently working and testing, projecting at the group level, for example, cortical thickness, gray matter. We used it recently to project PET, directly receptor PET onto the white matter and build the circuitry of the different neurotransmitters. You can do, I mean, like the principle is a weighted average onto the white matter of what you have in the cortex, and you can apply it to anything you want, any kind of modality, and run statistics after that. Yes.
Sorry, this might be a very stupid question, but if
Amazing.
we assess how much of the white matter tract is impacted, for example, or eventually the probability-wise, how do you assess how much of the?
Well, in that case, it's just a healthy control, so there is no damage.
Yeah, and in patients.
This connectome will be tomorrow. Like that's where we assess how it is damaged. All good? Cool. Well, I'll go through that extensively tomorrow, really. Okay, so I'm going to give you some example of this because then also practical how to do fMRI, you're going to do it with AV. Really is just changing the input of the analysis, and the rest is exactly the same. So we did, since we invented the method with Victor, we did a few studies that were more or less successful. So we worked on attention, for example. So you have here a paradigm that manipulates attention in different way. It is derived from the Posner paradigm, for those of you who know. Where you can manipulate the preparation of attention. So the task is essentially to fix, say, the central cross. And you need to detect whether you had a target that appeared on the left or on the right of the screen. And you can manipulate the attention of participants before the target appears to be able to assess the process that makes you faster to say it appears on the left or it appears on the right. For example, you can tell the participants the target is going to happen very soon by preparing them with a central cue that happen, with an alerting cue, which is a tone that happen exactly before the target, exactly at the right time before the target. So like the participants are really on edge to detect the cue, and they're faster to say it was left or it was right. This is preparative attention. Or you can give them some cue on the side where the target is going to appear by giving them mostly valid cues, usually 100%. So you say like the target's probably going to happen on the left. And so when it happened on the left, you're very fast to say, well, it was on the left. But when I lied to you and I told you it would appear on the right, and it happened on the left, you slower. And that is the orientation of attention. And then you can have like the selection where I give you a symbolic kind of cue that will tell you if it's green that it will appear on the left, and when it's blue, it's going to appear on the right. So you're going to use more cognitive abilities to convert this symbolic cue to make a selection on the side where you're going to make your detection. So we applied the functionnectome on this paradigm, and we've been able to find that you have some circuits that just come out as significant. For example, when you have to prepare your attention, you use a lot of the middle longitudinal fasciculus and the arcuate in the right hemisphere, and also the acoustic radiation, because you hear this tone. Okay, so that makes sense. But instead of having a single area of activation here now, we have the full networks that comes out. For some reason in that experiment, and that's why I say, you know, it doesn't work all the time, we didn't have so much results for the orientation of attention. I would have expected frontal-parietal connections, but that didn't come out. Is that a problem with the experiment, or a problem of the method, or a problem with the tractography? Was the subject not having the same connections because they're coming from Spain? I don't know. Just didn't work out. And when you look at the selection, that's what I told you, that requires a conversion of this symbolic cues. Then you activate a lot of region and a lot of network, including a lot of supernodal fasciculus, but also anterior thalamic radiation, corpus callosum for the integration between the two hemispheres. So you see how simply by transforming the data, then instead of having single region activated, you have a wall circuit, and then you can transfer this wall circuit into charts that I'll show to you after that to try to interpret the mechanisms that are behind in the brain, and then try to play around being like, so what will happen if I cut this connection in my system? Will I have a specific flavor of disease, or is this the entire function that's gonna fall down? So still with Ma, we've been exploring another question that I found way more interesting is like by manipulating the visibility of the target. So sometimes you can see the target, but sometimes the target is so dim that you cannot see it. And we play really at the individual threshold of people who can see or cannot see this target. And by doing so, we can make the difference between something that is perceived consciously by individuals versus something that is still perceived in the early processing of your brain, but will not reach consciousness. By doing the contrast between the two, you can have on one side the conscious experience as defined as being able to report what you saw, which is only one dimension of consciousness, versus early processing in the visual cortex that is not reportable because it has not been selected and identified by the brain as something that should reach or be manipulated by your consciousness. All good with the task?
You have to change your N, the number of participants with this sort of prior.
About the method? No, it's the same. It's exactly the same. Nothing to change, except putting it in the Functionnectome box and bringing it back into the processing.
Sorry, so even when they wouldn't see it, they were forced to choose between one side or the other.
Yes.
Okay.
And that's about chance. That's how we know that it's just like no, they didn't see it.
Okay.
We're good.
And how big was the sample size in this?
I don't
Roughly.
remember. That was
50, 100?
I honestly don't remember. I think it was like 30.
Okay.
Like a normal sample size fMRI study, not 100. Yeah, I think it was 30.
I was wondering if we don't have the diffusion image, since the tracks are subject specific somehow, can we also use alternatively some type of template in case we don't have that?
So the Functionnectome include anatomical priors of how things are connected together. You do not need diffusion MRI. It's completely bypassing the entire tractographic diffusion MRI to make it available, to make those questions available to people that don't do connections. And also it is based on the assumption that the average brain, which we all do by the way, the average brain is a good representation of the healthy brain. That's it.
But, so in this case, it means when everything is settled according to that analyze space, right?
Yes.
And can't be changed. So for
Yes, you need to be in the MNI 2 millimeters. That's just basically what you
So
know.
for example, fetuses and neonates in this case.
What?
For fetus and neonates.
No, we didn't do, we'll have to write specific priors for that.
I hadn't written that, but I didn't find the option to change this space, since I was wondering if there were some options in which you can select your space, in which you're working on instead of M9.
Yeah, no, so there is like, there's a few questions regarding that, which is like population specific templates that we don't have. For example, agents like to have their own templates. We didn't create that. We can't do everything. There is like a different age template, like fetuses, for example, cannot really use an adult brain priors for fetuses. You need to create your own priors, but it is possible and it is written in the manual how to do so. So we can totally do it. But you will need diffusion in that case. And then recently we spoke a lot with Robin because we wanted to look at brain asymmetry. This is a brain that is the prior asymmetrical. The brain is asymmetrical, but if you really wanted to test functional asymmetry, you will need symmetrical prior, which we also didn't have time to do. So one day we'll do it with Robin, but we never found the time. So a lot of work.
Can we define the probabilistic then? Because I mean, in the end, the results you got is based on your time series and on some checks that I've provided for average, you know?
Yeah, it's average connectivity matrices. That's more than just some tracks, but yeah.
Okay, so that's probabilistic, no?
So probability is at group level. So you know that.
Because you could obviously you can have as many subjects as you want. And
Yeah.
okay.
You good?
Yeah.
Okay, we can come back to this.
Yeah, thanks.
We're good? Okay. So what happened when we saw the target versus we didn't see the target? Well, if you look at the comparison between classical fMRI analysis versus functionnectome analysis, and you plot the Z values, according to deactivation, which is what is more activated when you didn't see them when you saw the target, versus activation, which is what is more activated when you saw the target than when you didn't see In some, you were conscious of it. You can say you saw the target. You were not conscious of it. You didn't see the target. You can see that you have what we call a double dissociation. You see in blue the classical activation paradigms. I will show you that you have very few voxels that are significant over here that are showing you voxels that are significantly more activated when you are conscious versus when you are not conscious. And also very few here voxels that are also significant when you were not conscious versus when you were conscious. Those voxels here are represented there, mostly in the intraparietal circles that light up full power into the classical activation paradigm when you're conscious versus when you're not conscious. When you look at the functionnectome, something interesting happens, which is you don't have that many interactions in the brain that are significantly activated when you're not conscious, when the information stays stuck into the first part of the visual system but never reach your consciousness. But as soon as you're able to report those stimulations, this target on the screen, then you light up full power, more than classical fMRI analysis for significant Z values. And those include a large network of regions. They include frontoparietal connection, posterior segment of the arcuate fasciculus, anterior thalamic radiation, and corpus callosum. Why is that? Well, the visual information stays stuck into the visual ventral field, visual ventral path. We get decomposed in those different modules that will tell you, oh, well, I've seen this kind of shape or bar like this, or those were letters. And then in order to be conscious, you need to be able to manipulate it with whatever you want to call it, working memory or a global workspace. To do that, you need to have a direct connection between this ventral stream and the frontoparietal network that will be also activated here, okay? Not to forget that part of being conscious and manipulating something is being able to verbalize it, which require connection with the other hemisphere, language areas. And that the thalamus, often forgotten, is the conductor of synchronization in the brain. So together now, instead of having the intraparietal circuits being the locus of being conscious in the brain, now you have an entire circuitry and mechanisms that are working in synergy that can create your conscious experience. In contrast, you can see that when you're not conscious, this phenomenon does not happen, and you remain in the very primary early processing of visual information that do not require interaction with so many regions of the brain, because it was not judged that as that important. Yes.
Believe that somehow, sorry, it will be possible to reveal the contribution of these regions that go beyond this very narrow cluster of activation. With fMRI, can we?
You mean like characterize them in terms of their specific functional contribution? It's a very good question. So, and that will be tackled on Thursday by Valentina Pacella, that will speak to us about the fabric of cognition. We are limited by the granularity of what we define by function. What you define as consciousness might be divided into further, more extensive division of processes for which we don't even have words, or we cannot even imagine what it is. Very limited by observational bias. I'll speak a little bit about that tomorrow as well. So far, behind these big words, you have all those mechanisms that we try to explain and put together. But the level of description of function as we know it requires interaction between several brain areas. Are those brain areas functionally specific, but we just don't know their function? I don't know. It's an unknown.
So in a way, if the thalamus is involved, we should be able to differentiate its activity between these two experimental conditions, right?
Yeah. Well, the thalamus besides probably the early visual input, didn't pop out as significant in the.
Yeah.
So it's not that easy. Yes.
Is there instead of using Functionnectome, for example, so the diffusion levels prior, for example, for the default network?
Yes. Well, you know, you usually use it as a data-driven approach. If you start using priors of the network you want to see and put it into your functionnectome analysis, then you're probably going to see what you put as a prior because it's not in competition with any alternative network that you would like to test. It is, yeah, it's a lack of control hypothesis if you kind of force it this way. Okay.
Just in order to be sure to understand, what you call voxels activated.
Yes.
In the right, right parts.
It's the number of voxels activated.
But you include the voxels that are seen in the white matter.
Yes. And if I had, very good question. What was your question, by the way? If I had more stuff, I think the food is here. Sorry. Thank you. If I had more significant voxel everywhere for the functionnectome, then will be, oh yeah, that's clearly because there is more voxels that are reported that can actually be activated with a mode. But the fact that we have a double dissociation is a demonstration that it is not the case. Lucky enough. Yes. Yeah.
Maybe I have a lack of knowledge of diffusion and right. But the thing is, I don't understand if the ROI of interest are defined when I define the effects, or the functionnectome is also able in some way, starting from the sine series, to kind of decompose the signal and identify the independent component and kind of merge, like a kind of integrated ECA analysis inside.
It's way simpler than that. So you take the bold signal of one voxel, you look at how it's connected with the rest of the brain, which is the probability that it's going from 0 to 1, and you multiply this value along the brain. And you do it in a normalized way so that the sum of the probability is equal to 1 everywhere, so that you never have values that are higher in the white matter than in the cortex. So I went a bit far there. What happens when things are connected together and vary together, they will have values that are projected and very represented into the white matter. You'll see the functionnectome is a more complex version of the disconnectome. So I started with the hardest part, then tomorrow I'll be like, oh wow, it's so easy.
In the figure, you displayed also the cortical voxels that are connected to the white matter.
Because the white matter is going inside the cortex.
So
there's no isolated island.
So that's a sort of enhancement of the only bold effect.
Yeah.
As you said, the visual word form of object form area is connected to the ventral system.
Yeah.
But we can't see that with the bold during your experiment. But
but we can see it here. Yeah, well, we're not getting rid of the cortex. The cortex is important. We're taking advantage of its connective property. So you technically not saying cortex useless, now I'm going to work on white matter. You change the functioning model behind the way you assess the brain. You change the assumption. You don't want to look at local activation. You want to look at coherence between brain regions that are connected, or voxels that are connected, because we don't use ROI. So.
Can we say that it's a way to overpass the limitation of the bold effect?
Yeah, I tried to say that, the reviews didn't like it at all. So we're like, you can't say that. You can't say that. The standard paradigm is still the gold standard. So yeah. I was like, no, okay. Yeah.
Does functionnectome allow, like if there's a data set that has both the diffusion weighted imaging and the functional imaging.
Yeah.
Is there a way to also
Yeah.
take advantage of that with the functional?
Yeah, that's what I mentioned before. If you do that, then you had the level of complexity, which is the entire individual variability. So you will reduce your significance at the group level because you're not forcing the same prior on everybody. And so they will all be different. If you want to do single cases, that's interesting because then you have the neuroanatomy of your participant. It is possible, it's in the manual. It is very time consuming, but you can do it.
Yeah, so I understood that the prior that's incorporated is the second.
Yeah. What you don't want to do is do a group level analysis with the individual diffusion weighted MRI of your participants to study the group level results. That will make no sense because you're trying to use individual tractograms to find what is common between everybody. What I already give you what is common between everybody in terms of tractograms.
But wouldn't there be a way to even if it's like individual tractograms, maybe there's a way to register them all somehow into a more.
Can't fix different phenotype. You can fix eventually misalignment, but you have people that are fundamentally different. Left-handers, right-handers, for example, it's like, yeah. Okay. Ben Penfield showed it for the area for language. And the old papers he was stimulating, he was showed like how two patients have different mapping of language in their brain. You need to choose. Yeah, either go single cases, individual subject, maximum resolution, understand how this person works. Or you go group level average, and then you assume that the mechanisms are similar. And then you can use an average prior.
Still wondering if there's a way to take that into consideration, like which person, because Stephanie was mentioning before, you can predict kind of the recovery of a person already.
Yeah.
Take the
But
volume of the.
in that case, you do single subject analysis. Or your variable, you cannot have two unknown, then you cannot disentangle them, you know. Was your difference in the functionnectome due to the difference in the white matter, or was it different in the way that white matter was used?
Still, maybe there's some overlap within even with the individual papers that can.
You can play.
Extract something at the group level.
You can play.
Yeah.
But usually tackle one unknown at a time. Otherwise, you can get lost on the way into multiple possible interpretations. But I'll be happy to, if you find a solution, I'll be happy to read it. And so this allows you to, you know, start thinking about models where you don't only have region, but also the pathways that you can put inside it. In the present case, we were saying the visual input follows the visual ventral stream, and you're not conscious of it until you get into the ventral lateral temporal cortex that is connected with the IPS, with the posterior segment, that will bring the information into the frontoparietal network over there. And these synchronizations are conducted by the thalamus, that without the thalamus, you have an orchestra of regions that are not perfectly synchronized. It's here to fix the tempo. And then you can also send information into the left hemisphere in order to be able to verbalize it. Interestingly enough, and we'll speak about it a little bit tomorrow, you have cases of patients that can have a disconnection over here and present surprising behavior of visual neglect that was originally described by Halligan and Marshall. If you have a lesion that disconnects the ventral temporal cortex and the intraparietal sulcus, patients, we will show the same house up and down, except that on one of the house, the left part is in fire. We'll tell you those two houses are the same because the visual information from the left visual field didn't reach the frontoparietal connection that allow to manipulate the fire representation. However, if you ask them which house they would like to live in, they always choose a house that is not on fire. And you ask them why, they will be like, because it's a nicer house, got bigger windows and look more spacious. It is exactly the same. So that's a form of confabulation. So the interpretation that we had is that the information of the fire is not reaching this working memory, global workspace manipulation. So you cannot be aware of it. But it's still reaching early processing and emotional feelings that something not right. You have this priming of one house is better than the other. Because your frontal parietal network is intact, it builds this justification of why you should be in this house. And that's a typical confabulation. On the other hand, patients that have a lesion of the frontal parietal network, they won't see either, they won't be aware of the fire inside the left part of the house, but they won't have any difference in the way they choose the house. It's like they cannot confabulate about it. Okay. We'll tackle that with disconnection, introduction to the disconnection tomorrow. So you can do tons of things with the functionectome. You can also assess functional connectivity and the decomposition with independent component analysis of the functional networks that you have here in different resting state networks. And so that's a trick that we did. We did the ICA at the same time on the functional network, but also on the functionectome. So we did on the classic and the functionectome version at the same time. And the ICA decomposition give us together two maps every time. There were the connectivity in the cortex, as well as the subcortical connections that are covering with this connectivity in the cortex. And the result of the decomposition were actually nicer than if you. And we produce this white rest atlas. And then this is another example, and I'll speak more about it tomorrow, where you can project directly if you use large database meta-analysis of fMRI with different tasks that have been studied over the past 20 years. You can do it systematically and project here for each part of the white matter what is the most likely functional term that would be associated to this white matter pathway. And you create those fancy neo-phrenologic maps where you can put name of the white matter. And of course, I will encourage you to realize this is just the top of the iceberg. This is the most significant term. Behind that, and we'll see that tomorrow, you have tons of other terms that also load on those regions, because there is not an equivalence between one single tract and one single function. It's more a pattern. The functionectome was coming with some limitations that we further tackled and improved in terms of priors. So for example, with tractography, we're not so good to get inside the cortex, but the white matter and the axons are going inside the cortex. So typically, the tractography that we have is like the blue part of the line, eventually going a little further. So what we did is we did a first step of extension of the tractogram so that they reach the entirety of the cortex. And so we can capture in the projection of the functionectome and the fMRI a maximum of signal from the cortex that we can send to the white matter. So we twisted a little bit our priors and our tractography so that we get full blown inside the cortex. And then, because we didn't want it to get outside of the brain, we also like shaved what was going too far. Okay. Another limitation that I mentioned just before is that you do your functionectome analysis, and it's a little bit like behind your TV when you have many appliances between, you know, video games and I don't know, like Nintendo Switch, I mean, this Google Chrome app plus the modem, whatever you connected to your TV, and then you move from one house to another and you get all these cables. It's like, I don't know what's connecting what anymore. It's a little bit the same with the brain. You can get a result and everything is intermingled. You would like to see that connection, but you're not really sure because there could be like two connections that are parallel to each other, not connecting the same thing. So we thought maybe we can clarify a little bit the result by disentangling the connectivity and creating different set of priors that are either association fibers that are connecting up to you. Different cortical ridge within the same hemisphere. Commissural fibers that are connecting, of course, and projection fibers that connect the structure with the surface. Exactly. Let's get back to our friends pushing that button. You can see now that you can make different maps that correspond to those different systems. Association fibers involvement when you push that button versus commissural involvement when you push that button versus projection involvement when you push that button. And then you can also compare the original analysis with the extended version of the tractography that I showed to you. So you get more information from the cortex and you end up with a result that is slightly more significant because you got more signal. And you can also use the three priors that you had just before that I showed to you to create those Z-max map, which is just showing you the maximum Z value in a single map of the connection that I involve combining commissural, projection, and associated pathways. Okay? So just small improvement. Yeah.
Is it normal if the task is supposed to be lateralized to one hemisphere to also get connections from the other hemisphere?
So you have, yeah, because you usually don't have tasks that activate only one hemisphere. You sometimes have a few voxels on the other hemisphere. See, typically you do finger tapping, you're gonna activate with your right hand, you're gonna activate a lot your left hemisphere. You have a little bit of mirror activation in the right hemisphere. And this has been showed in a paper by the Gene when they did it in hundreds of people, right-handed and left-handed. I'll speak about that tomorrow. You can study also the thing like this is working memory, where you can see that working memory mainly involves frontoparietal connections. This will be the original analysis, the analysis with the extended streamline that we see did not improve so much as the analysis. But when you split associative projection and commissural fibers and you do a Z-max here, you see way more significant results. And that is because frontoparietal connection cross with a lot of commissural and projection fibers that will dilute your signal. And Victor went a little far with this, but I think it's great. We don't ask ourselves the question enough whether short connection, medium connection, or long connection will have different contributions to your function. So sometimes you want to disentangle, local connectivity from mid-local to long-range connectivity. You can also do it. We have priors for this. Basically, you can start your analysis with some hypothesis about what you want to see and what you want to compare. Don't know the priors that are appropriate for it, and then just observe the results. Okay, tons of different maps, but not yet fetish map or symmetrical map. One day.